everyone, and welcome to the Bridge to the Bay with Dr. Veras. I'm Dr. Matt Veras, and this is episode 3 with Dr. Graham Ruby. Graham is one of my new coworkers, and I've had an amazing time getting to know him and working on some projects together. It really is incredible, the type of people at Calico, and Graham really exemplifies it. Now, since he's a professional scientist, we talk about what it's like to run a lab. But really, we talked about his journey to get here. In his training, he switched fields multiple times, which is quite strange in science. And we also talk about the arts, music and literature, and how that can actually help the scientific pursuit. We seem to both think that when scientists use both their left and their right brain together, that's where truly exceptional science occurs. And so, if that sounds of interest, enjoy listening along. If not, hopefully one of the future episodes will be of interest. Hey, Graham. Good morning, and thanks for doing this with me.
Absolutely, Matt. Great to see you.
Awesome. So I wanted to have this conversation, and thank you so much for agreeing to do this. Just for the people listening, we're speaking to Dr. Graham Ruby today. He's a coworker at Calico, a principal investigator, so he runs his own lab. And so I'd like to talk to him about kind of what he does and how he got there, because it's been quite a long journey along the way. So would you be able to just say sort of broad strokes what it is you do as a leader of a lab?
So it's basically I'm a scientist and biologist, and I am primarily focused on data analysis, so like bioinformatics, and that can mean a lot of different things. So I do some genomics, I do some physiology, some like just sort of biostatistics kind of stuff, genetic analysis, and as a group leader, I sort of also organize projects and have some managerial responsibilities.
Right. And so you're a scientist. Did you always want to grow up to be a scientist, or was that something that got stimulated later? When did you kind of notice that spark, if you can remember?
For the most part, I mean, one thing about me is that I have type 1 diabetes, which I got when I was 9. And that was, that was a bummer when it happened.
Yeah.
But it was sort of like an early exposure to, because, you don't learn about physiology really usually, when you're 9 years old.
Yeah.
But, you get a disease, you learn a little bit about it. And even though it was a bummer, it was also kind of interesting. And it gave me, I think, sort of an early appreciation for the power of like basic research, basic understanding of how biology works, how that can, have an impact on people's lives. And so that really definitely got me interested in biology and science. And I was always kind of a science nerdy kid anyway.
Yeah.
But I was also, I mean, in sort of, as a kid and sort of in high school, I was really more into the arts. I was more of a sort of music and art nerd than I was a science nerd.
Yeah.
And that sort of switched when I went to college in a lot of ways. But what I really didn't appreciate about science in high school or in the early years of college, but started to appreciate more once I joined a lab, was how creative of an endeavor science is.
Yeah.
And, when you first start doing science, you're doing a lot of sort of memorizing facts because there's such a long history of amazing discoveries that have been made about the universe. And you learn so many fascinating things, and that's fantastic. But you don't really sort of approach it in terms of sort of charting new territory and having to be creative about how you would design an experiment. And you
Right.
can do sort of science fair projects, but at that point, you're not really entrenched enough in the science to really be super creative,
Right.
or do something that's really, sort of at a cutting edge of getting into a question that nobody knows the answer to. Usually
Yeah.
work on a question that sort of you don't know the answer to because you're just in like middle school or high school, right? So you like there's a limited amount of stuff that you know, and
Right.
you have sort of limited skill looking things up at that
Hmm.
point. But but yeah, you uh, once you get more into science, it really is a sort of a creative thing, sort of designing experiments. It's sort of like its own special kind of art project.
Yeah.
It has some more constraints than usually in art. I mean, artists have to deal with critics, but to some extent they can just like ignore the critics.
Yeah.
The sort of the scientific method is sort of like an abstract critic that you can't ignore.
Yeah.
But but it's also that science involves a lot of writing, which
Hmm.
was something that is not a big part of science curricula up until
Yeah.
you start to get more advanced. And so
That's
I
true.
actually, thinking back to my high school education, like I said, I was more into the the arts and everything, and the AP, and sort of more advanced classes that I took were more sort of literature,
Hmm.
and I was really into writing and sort of
Oh, that's good.
emo poetry and all that. Um, but I really felt like that aspect of my high school education was what really prepared me for science more
Yeah.
than anything else in high school,
Hmm.
because there wasn't really, also like in the college science curriculum, you start to get to the point where you have to do a lot of writing,
Yeah.
but no one's teaching how to do good writing,
Yeah.
right?
Yeah.
They're just sort of criticizing it for being bad if it's
Yeah.
bad writing.
Yeah. You learn, like, get thrown in the fire, and you learn by osmosis, I guess. Right.
Yeah. Whereas, I mean, in high school, you're doing all these things about like, 5-paragraph essays, and like,
Yeah.
here's how you introduce your point, and then here's how you support it and summarize it, and all that sort of stuff that's really just about writing
Right.
becomes relevant to science, but at this later stage.
Yeah, that was the stuff I didn't pay attention to in high school because I
Hmm.
guess they couldn't articulate the utility. And then it was kind of like toy writing, like it was very prescribed.
Hmm.
And I found I wasn't allowed to be that creative, at least in
Hmm.
high school.
Hmm.
And so it put me off. But I had to learn a lot over again when I got to grad school. Even at that point, like
Yeah.
I could put together, like the basic science report. I could write like a lab report, that kind of thing. But it's very structured and it's kind of prescribed. You have to get a certain amount of information in, and that kind of fills the page.
Yeah.
But once you hit grad school, there's different types of writing too. Like you got to write scholarships, and you have to start tell a little bit of a story. And then once you hit a manuscript, like a paper, it's really a large undertaking. And if you don't synthesize it right and tell the right story, it's not compelling.
Yeah. Yeah. Definitely. So yeah, it was, and my high school had a really strong sort of like literature department,
Oh, that's great.
and it was really the one of the things that I love, because I'm a huge Star Wars fan,
Okay.
which is too much of an aside, but the curriculum at our high school really focused across many years on sort of like Joseph Campbell and the hero journey,
Hmm.
and sort of thinking about things archetypally, and that
Yeah.
sort of that, I mean, that plays into science because it's a little bit different structure, but you still have that same structure in a scientific story where you're sort of called to adventure by some question that you don't know the answer to.
That's a good way to put it.
And
Yeah.
yeah, and then you go out, you do these experiments, right? And then you get this result, but then the
Yeah.
result doesn't stand on its own. You sort of have to come back to that world and say, well, here's how that fits in to this broader literature that I was summarizing at the beginning. And hopefully it does have some
Yeah.
impact or interaction with that.
Hmm. Yeah. So, yeah, you talked. It sounds like you really looking back could take something from your high school experience. We go back to like where did you grow up? Like how did this all get started?
Yeah, I mean, I grew up in Southern California,
Hmm.
in Irvine. I went to a Catholic school, Santa Margarita Catholic High School,
Okay.
in Orange County.
Yeah.
And yeah, it was uh, it was the head of the English department there was a woman named Molly Merrill, and she was just fantastic. And I had a number of good teachers, but one of the things that was sort of, I think, unique about the school was that it wasn't. I think the year I graduated was like the 10th anniversary of the school opening,
Oh, wow.
and so she had been like the first department head. She had started it like not too
Hmm.
long ago,
Hmm.
and I think that sort of, there's certain things that you sort of have to do in a high school curriculum of any type,
Yeah.
but and then some things sort of historically get sort of baked in.
Hmm.
And so, since she had to set up the department and its curriculum so recently, it still had this cohesiveness
Yeah.
of, like I said, the sort of like mythology, archetypes, hero's journey sort of stretched throughout. It was like a running theme throughout throughout the whole curriculum, and not just the main courses, but the sort of more elective type courses.
Yeah.
And so that was really, really fantastic. Uh,
Yeah.
I mean, the other thing that I started doing at that time was playing guitar,
Hmm.
which I still do today. And I took lessons sort of starting in my freshman year of high school,
Okay.
and I had a really great guitar teacher. He was just When I met him, he was just the teacher at like the local music shop, but a guy named Carl Arano.
Okay.
And he had been a student at Berklee College of Music in Boston,
Wow. Yeah.
and he really, his sort of curriculum that he followed for students, to the extent that he could, right? I mean, that's one thing. Most kids learning to play guitar want to learn how to play like these three Metallica songs, right, at that time.
Yeah.
And but I was really excited to learn about different types of music and music
Hmm.
theory.
Hmm.
And so he sort of took me through that curriculum, and it was, I think that was another case where it was sort of eye-opening about how a good curriculum and having things in context really helps you to make progress quickly.
Yeah.
Um, so I it was something that I took sort of with me to going forward into science, and probably talk about this a little bit later, but
Hmm.
sort of within biology, I've moved around a lot in terms of the different things that I've studied.
Yeah.
And I always sort of think back to those sort of early lessons with him, because coming into high school, I had a couple of friends who were musicians,
Hmm.
and they were like already really good. Like
Yeah.
my friend Josh was like a child prodigy on the piano, and my friend Jason was like a really good drummer.
Yeah.
Like already, or at least I thought. I mean, actually, by the time we graduated, he was like a fantastic drummer. At that time,
Yeah.
he was still learning, but I
Yeah.
thought he was just like a fantastic drummer.
Yeah.
But sort of like being around them, and hanging around them, and they were like, hey, like you should learn to play an instrument too, so that we can like play in a band. And
Hmm.
I sort of felt overwhelmed. I was like, I'm so far behind. Like
Yeah.
these guys are like, they're already experts,
Yeah.
and but nonetheless, my parents really encouraged me to sort of, you know, pick up the guitar and take lessons, which I think, for the guitar is a little unusual, for parents to encourage it, but they encouraged it, and and I took these great lessons, from this really good teacher, and it was sort of, by the, even by like junior year, I was a pretty good guitarist. And it's sort of,
Nice.
like by senior year, it was sort of seemed ridiculous that I would have been worried then about like, whether I had started freshman year or if I had started in middle school or in elementary school, there's
Mhm.
no, like, it doesn't, once you sort of learn how to do something, that doesn't matter. Those early huge discrepancies sort of disappear.
Right.
And that's something that I tried to take with me into science in terms
Mhm.
of moving into new areas and trying to learn new fields or new skill sets.
Yeah, I love the way you put that, and that mindset of uh, learning how to learn, first of all, but
Hmm.
understanding that if you have context and a direction, you can make the learning more efficient in some ways.
Yeah.
Um, and I love that you're likening sort of the arts and the creative process to science, because I feel like general public doesn't equate the two. I think it's sort of, people say it's like left brain, right brain. The types of people that do these things are only good at one or the other. But I personally believe that they feed each other, and that, just like you said, that creativity is important. It's almost the interplay between brain hemispheres that really can create that spark where you can do something truly novel, and game changing, really. Um,
Yeah, and I
but,
mean, and it's also striking me though how sort of sequential it is though, right? Because
mhm,
that sort of left brain right brain stuff, like when you're first starting to get into science and starting to learn about it, like that does apply. And
mhm.
in college, I have to admit my intro bio class grades weren't that great, because
Same.
it's a it's a ton of memorization.
Yeah, I didn't like
And
that.
yeah, and it's sort of uh the um sorry, I'm stuttering at this, but the early stage, there's not really that opportunity
Yeah.
to be creative until you've sort of learned that language and acquired that context.
Yeah. Yeah. It's very much, you got to learn the basics, and it's a lot of history, like you were saying. But that's what allows you to stand on the shoulder of giants when you really continue on. But you got to build on this immensely beautiful work in many cases, right?
Yeah.
My main issue was I didn't get the context, why I was
Mhm.
learning it, why it was important. And it wasn't framed in a way that I could understand that scientists that made this discovery, what was going on in their life, in the world, that even allowed that type of thinking to be possible.
Right.
Because I think that really frames it as, a story and something that we can latch onto as people. We can kind of internalize when it's a story about someone, and we can put that whole sort of picture in our mind. It allows more connections so that you can pull on it better, from memory.
Yeah.
And I think that it's a shame in many ways that they're kind of forced to do that, in university, just because there's so many people, and
Hmm.
it's difficult to evaluate 1,000 kids all trying to learn the same thing, and you have to screen them through as well to some degree. But for those that really just want to learn, and that you know don't love the memorization, I think it can turn a lot of people off.
Yeah.
And
Al- Although I
yeah.
do think that's sort of a necessary hazing, right? And I think I mean, it would be nice if you didn't have to do that, but I'm just thinking as an undergrad, I was at Northwestern,
Mhm.
and worked in a great lab that was really really welcoming to undergrads, and really was tough on us, but sort of encouraged us to really really dive into research. It was
Mhm.
Rick Morimoto's lab, and it was a great environment. But there was this thing of, when I started working in the lab, I'd never really read scientific papers before. And
Right.
now I'm, being handed these scientific papers, that are relevant to the project I'm going to start working on.
Mhm.
And, you only have a little bit of context for any biology, I think, as an undergrad when you join a lab. Like, you're not
Right.
like, oh,
Oh
I
yeah.
want to study this. What lab studies that? And you go find it, right? You're like, what lab seems like it would be a cool place where they'd welcome me and I'd want to do
Right.
research.
Yeah.
And then it's like whatever they study, I guess I'm going to study that,
Yeah.
especially, and it's like, what project I'm going to do. It's like, well, whoever needs help, I'm going to help them with some sub project that's related to their project. Right. You're not making that choice. But I guess, so first getting into that and first starting to read scientific papers. At first, I just had to read them on sort of a blind faith because, especially in molecular biology, there are so many acronyms.
Yeah.
And that's sort of, to me, that was the big language barrier. And I would read these things that I would have no idea what anything was referencing. And that was the sort of the same memorization aspect that I had the most trouble with in sort of the intro classes.
Yeah.
But then, you read more and more, and, just like speaking a new language, you start to recognize more and more terms. And then at that point, you can start to think about, the sort of the logic of it, right? The players. It's like watching a TV series, right? Like in the first episode, there's a lot of mystery because you don't know who any of the characters are. And then later on, and, you get into things, it's probably for like Star Wars, like it's, people who aren't big Star Wars fans, like I don't have this problem, but I have more of this problem with like the Marvel cinematic universe where it's like I haven't seen all the movies, so I don't really know all the relationships between all the characters, and so I can get a little bored with it and just like whatever. But if you're into it, if you've seen all the movies, then there's a lot more to draw on. And you're like, oh wow, that's amazing what just happened. But you really can't get that out of it until you know all the characters and all the players and you understand their relationships. And that just, like you said, you're standing on, the shoulders of giants and centuries worth of giants.
Yeah.
And until you build all that up, it's like, jumping into a soap opera in the middle of it, you're just not going to know what's going on.
Right.
And it might even seem kind of silly, right? But it's not silly. It's good stuff.
Yeah. Yeah. I love the way you're sort of speaking about it because you're speaking in artistic terms about science, about the scientific process. I just want to, talk about sort of that hero story from high school. First of all, it's nice that at that department chair could have a sort of a unifying vision that she wanted to pursue and felt that it was important. But I think this sort of hero mythology applies everywhere, and it's something really central to how humans conceptualize the world and their place in it. And for me, I don't have a problem with it. I'm unashamedly cheering for the hero always. I try and see myself that way, and that I have to worry about the cockiness aspect and that kind of thing. So make sure I have friends that don't let me get too arrogant, things like that. But I think this story of the hero is useful for everyone because that's the point, right? It's achieve or strive for some sort of ideal, to be the best of yourself, to do something extraordinary. And I think that's worth pursuing. It's an inspiring story. Do you see any scientists in history that you look of that way? Do you think that applies to scientists and how they pursue their endeavors?
Well, I mean, so the hero's journey, this sort of cyclic pattern, and for anyone who hasn't heard of this before, sorry, I probably should have said this earlier on, but this guy Joseph Campbell, he studied world mythology, and he sort of, as he read more and more myths, he sort of noticed that there was a certain pattern of events that was in common to all of these stories, with people sort of, they're living their lives, and then they're sort of called to adventure. Yeah. Then they enter another world, and then they're sort of helped out by this person or that person, whatever, and then they sort of reach some sort of realization, and then they have to return to their original world, but as sort of like a changed person. Yeah. And it's sort of, it's a pattern that once you sort of know what the pattern is, once you've, read that, then you read almost anything that people like, and you can see that pattern.
See it. Yeah. It's everywhere.
And that's one of the great things about, one of the things, I mean, Star Wars in the 70s when it came out
So
was like,
perfect.
it was like really cool, like cool special effects, but it's not the only, it's not the only like cool space movie. And there are a lot of like silly things about it,
For sure. Oh yeah.
But at the same time, it's like I think part of the reason people love it is because like George Lucas had sort of read this theory of myth building, and it's like very literal. Like each step is like in there in a very literal way, and so it's sort of it really plays into this. And I guess the idea is that it reflects sort of how we all grow, like how we all sort of grow through life and all of the challenges that we have, but also sort of describes the way that we think about our own successes. And so it's sort of just like a description of what we do psychologically as we live. And so having that, so certainly having that framework and sort of already knowing that, okay, there are these certain steps. If you want to write something that's going to resonate with people, it makes it easier to just already have that as a template, right? I think that's a good way of thinking about the scientific method. And when you learn about it, it's very abstract, right? And you're sort of, when you do like the, middle school, high school, like science fair project, it's like, you have to say, this is background research, hypothesis, test, data analysis, conclusion, right?
Yeah.
But those really are sort of the the steps. Like that's sort of the science version of the hero's journey. And a lot of times, I think scientists like us or people we know will sort of say, that's not really how it works in real life. It's like, you do this experiment, but then you mess it up and then you got to go do something differently. And then you sort of notice something strange and sort of in retrospect, you're like, oh, I guess that's what I was really testing. And, the way that you write up the story isn't always the way it happened in the lab.
Yeah.
But at some level it is, right? At some level you had some set of things that you knew.
Yeah.
You had a thought, a hypothesis that you wanted to test or a question that you wanted to gather more data on. So you set up an experiment, and even though it's messy, I mean, life is always messy. And, the hero journey, sometimes it's only in retrospect that you sort of see, oh, these are how I went through these steps as I went through.
It's not linear.
As I went through. Well, but in retrospect, if you go back and you say, oh, as I think about this process that I went through, like it's a lot harder to say I'm at this point in the process than it is to say a year later, oh, at that point I was at this point in the process. Right. And I think the same with science, where really that's the scientific method is, it's not just I mean, one way to think about it, and I think this is a good way to think about it, is as an effective technique for gaining new knowledge that is going to actually be legitimate and useful.
Yeah.
But the other way to think about it is just as a description of the process that anyone who's investigating the universe is going to go through. And sort of knowing those steps allows you to sort of execute that more effectively, just like George Lucas writing Star Wars, just following the steps of Joseph Campbell. Is a lot easier than just trying to come up with a story and refine it over the years so that it resonates with people until you get to the point that you sort of arrive there by accident.
Yeah, and there's some sort of core messaging that people have always responded to in that hero mythology.
Yeah.
And I agree, Star Wars just nailed all of the archetypes. It was so beautiful, right down to the Force, which is sort of like this magical, existential sort of power that certain people have a certain acuity for, or, they can shape things how they want. And I think that's a great analogy for life. There's certain people that just have certain insight into what it is to be a human, or what motivates people, or what is that unifying vision that's required for people to move towards a common goal. And it's those people that can rally a movement behind them. And that's probably exactly what George Lucas was doing when he was setting up the film, even setting up the cast and crew. It's a huge undertaking that was years long in the making. And it was such a complex fictional world. It required a lot of moving parts to put together. And so it required some sort of unifying vision and some passion for people to use to strive toward that amazing product in the end. And you can kind of feel that magic that people put together, right? When people really care and are passionate about what they're working on, that pursuit of their ideal is recognized, even if it's through multiple layers of screens or whatnot, or even fictional layers of story. It can still resonate.
Yeah. No, and I mean, I think the other thing is like the power of that sort of the underlying narrative.
Yeah.
And, and sort of the fact that he has that narrative in there. The same with the scientific method, right? Like the great leaps in science, you can sort of, no matter how intentional or unintentional it was at the time, you can see the steps of the scientific method. And I think, the same thing, and it's like different people have, like, there's some really silly things in Star Wars, right? Like some people don't like the Ewoks, some people don't like Metachlorians, some people don't like Jar Jar Binks, and yet, like, even going through that sort of prequel era where people were, I think, at the highest extent, like, what, like, like we all sort of loved it still. Like, it didn't stop us from loving Star Wars, and I think that's if that deep story is there, that substance is there. You can like the minutia. There's still like lots of cool special effects. You can not like it. You can be like, why are mitochondria now, controlling the force? Like, that's stupid. Yeah. But regardless of that sort of surface stuff, you've got like that deep, that sort of like the Chomsky universal grammar, that's sort of deeper than the surface structure that sort of keeps you, grabs hold of you.
Yeah, I think I've heard Jordan Peterson speak, I think, the most about this sort of idea of like sort of a metastructure being more real than our reality, because it informs all of our decision making and our goal oriented behavior. And it's you can deny it all you want, but you end up acting it out anyway.
Yeah.
Yeah.
This is getting deep.
Well, that's what I hope to get from a podcast and a longer conversation, right? It's I don't want to just talk about science with scientists. There's some really amazing people that I work with here that didn't make their decision to get here, or didn't take it lightly, right? And it's informed on broader perspective and knowledge, and I want to get at that. I think that's really important, that the general public doesn't just think of science as some sort of sterile, very regimented thing. There's people that are contributing to our collective knowledge, and you have to do that as a human. Like, you're not a robot, and so you bring in your own stories and motivations as well.
Yeah.
And to that point, like, I definitely experienced scientists that don't want to think of anything broader than what they're working on, and I didn't really jive with those people. I felt like they didn't have a context that could inform their decision making, and the details or their actions tended to be cyclical or not moving in a productive direction, in my opinion.
Right.
Do you ever find pushback? Or I don't know if you talk about this with colleagues and things like that, but do you ever find pushback on sort of talking about the scientific method, or how you go about things in more sort of creative, poetic, artistic ways, or creating assimilations?
I'm not always this like poetry focused when I'm actually doing the science. So yeah, it's I do think that a lot of the work, you're sort of, these are like the the sort of the the ideas that are up there, but you're not necessarily explicitly
Of course.
describing them at the time.
Yeah.
But there is, there is a this phenomenon that you brought up of sort of people sort of sticking in their fields and sort of not necessarily having a broader context, and that is something that is, I think a challenge within science, and on a more practical level, I think a lot of that has to do with sort of the way funding is done and sort of the reflections that has on culture.
Yeah.
But one thing. So in a lot of ways, my career has been like pretty much like just the straight and narrow path, right? Like I went from high school straight to college, majored in biology, went straight to grad school, Ph.D. in biology, went straight to my postdoc, and then went straight to my job at Calico. I'm not one of these people who has like a fascinating story where I like went out and toured the world, or was like a medic in some remote war torn era or something like that. It was just really sort of following the steps. I think one thing that I did more than what I see in a lot of my colleagues, though, is changing fields and sort of getting into new areas of science, which has been really gratifying for me and had made me enjoy my career. I'm not sure it's the smartest plan, though, and I guess thinking about and this is something that I sort of thought about looking at the labs that I was in, and sort of as I started to understand a little bit more of how science is funded, where sort of like grants really go to experts in their field,
right?
And people who are changing fields when they first change are sort of by definition not experts in that new field. And my and I saw this with and just like I was saying with like guitar, where like 3-4 years after I'd started learning it, it's like it really didn't matter that I started freshman year of high school versus if I'd started when I was
right.
5,
Yeah.
But my PhD advisor, so I went to MIT for grad school. My PhD advisor was Dave Bartel, and he's like a very sorry, go ahead.
I just could you maybe just touch on the decision making that took you from sort of Northwestern studying biology and how you made that decision to go to MIT? What were the factors you were considering?
Oh yeah. So absolutely, and that's sort of, I think that's going to feed into this other story about changing fields too, because so like I said, so I was working in this lab that worked on cellular stress responses. And that's a fascinating field, and it's like it's something that sort of comes back in aging biology. But like I was saying, like when you pick or get picked to join a lab in as an undergrad, it's not like you're thinking about here are all the different areas of, like, you don't even know what many things are in biology, right? You're just sort of going with the opportunity. So as my undergrad advisor started encouraging me to to apply to grad school, and it's like you're getting ready to write applications, and you really have to say something about what you want to do and who the people in this department you want to go to, that you want to work with, like who they are. And so at that point, I sort of I had this this sort of this thing that I was thinking about, which was like a very sort of abstract concept that it was just something that I was curious about on some level, and that was sort of just sort of how sort of evolution works at the molecular level. And so and it was sort of just thinking about, like, well, okay, like, because most of what I'd learned about that up until that point was about sort of genes duplicating, and that you sort of, the way that you get sort of new genes in the genome as species evolved, is that like some old gene duplicates and then they can go do something different or, whatever.
Yeah.
Since it's just an extra copy, but you end up with something that's similar. But that sort of really made me curious about, sort of closer to the origins of life, like
Yeah.
how different molecules evolved, and did sort of each type of molecule or domain that's out there sort of have to spring up on its own, or is it easy for them to sort of to change,
Yeah.
like we always think about them sort of maintaining their basic structure and changing their function somehow, by changing certain properties of like what they're going to interact with, or binding pockets or whatever. But those big leaps, how do those happen, and are they
Yeah.
really so different from the small leaps? And
Mm-hmm.
as I started reading about that, I was really excited to
Yeah.
see that like there actually are people who study that sort of thing.
Mhm.
And I hadn't done a ton about it, of reading about it at that time, but the one of the most what I thought at the time, but I still think one of the most interesting cases that I came across of people studying this was Dave Bartel, who ended up being my PhD advisor.
Mhm.
And there was a postdoc in his lab, Eric Schulte, who had done a paper studying. So first of all, Dave came from a background of doing sort of RNA enzymology.
Okay.
And so a lot of biologists don't even appreciate that RNA, in addition to being sort of a messenger and sort of playing a structural role like it does in the ribosome,
Mhm.
can be a catalyst. And there aren't a ton of enzymes where RNA is actually the component of the enzyme that's doing the catalysis, but there are a few
Okay.
that are sort of around in modern biology.
Could you give an example? Would I know of one?
You definitely know of one. Well,
Okay.
I'll start out by saying a lot of them are play roles in virus replication. So if
Okay.
you have an RNA virus that does like a rolling circle replication,
Yeah.
then each new copy of the virus has to get cut somehow.
Yeah.
And in a lot of viruses, that's done by by an RNA that can sort of fold up and self cleave.
Interesting. Okay.
Um, and this was a contentious point for a long time, but now it's resolved. The one that you definitely know the most about is the ribosome,
Yeah.
where it is actually the RNA that's doing the catalysis. And
Yeah.
even though it's a big structure of RNA and proteins, and and like I said, that's well established now, but was very contentious for many years,
Mhm.
for many decades even. So and we might come back to that. That makes sense, but the sort of there's also a lot more, and this is what my PhD had worked on, in advisor had worked on during his PhD, was you can use in vitro selection methods to evolve new RNA enzymes out of random sequence. And so that was his PhD project, had been evolving an RNA ligase out of random sequence.
Mhm.
And that was sort of really getting at like origins of life idea. So there's this idea that since RNA can be like DNA, right? In a lot of viruses, it is the genome,
Mhm.
coronavirus, right?
Right.
Um,
Perfect
but
example.
it also can be an enzyme and can perform functions. So the ribosome is an RNA enzyme.
Mhm.
There aren't very many other sort of RNA enzymes in biology.
Mhm.
There's one in particular that's sort of also like a a core essential function, which is RNase P,
Mhm.
which is it cleaves the sort of it's like the 5' tail off of a tRNA from its primary transcript. And there it's like a cleavage. That cleavage of the RNA is being done by RNA. But
Interesting. Okay.
just like it's not an RNA just floating around on its own. The it's also in complex with proteins, and so it took some work to figure out that it was that it was RNA doing the catalysis.
Okay.
There's also a set of introns that are called self-splicing introns, because it's the RNA that does they splice themselves out of RNA. So they do all the catalysis. And then the spliceosome itself, again, this is a case where it was a little harder to figure out that it, establish that it really was the RNA doing the catalysis. But that's another case.
Yep.
So you have all of these sort of very like core function, sort of fundamental, like the most fundamental aspect of biology, of like making proteins, right? Where RNA is doing several of the catalysis steps and is doing the most essential of those for translation. The actual forming of peptide bonds is being done by RNA.
Yeah.
And so since RNA can do sort of both of these things, there's this idea that before life evolved sort of a different genome that's a little more stable than RNA to sort of store the information, and then evolved a different polymer system for molecular machines and for catalysis of proteins.
Yep.
That it was just RNA. That RNA was the the whole game, and that's called the RNA world hypothesis.
Okay. Yeah.
And so what my PhD advisor had done for his PhD is selected out of random sequences, just random RNAs, come up with a selection scheme to select an RNA that would perform a ligation onto the end of itself.
Okay. Wow.
And the idea there is that sort of a ligation of two RNA polymers, that formation of that phosphodiester bond, that's like the basic unit of transcription. That's what you're doing each time. You're taking a nucleotide and a string of nucleotides, and you're just joining them together.
Yeah.
And so that was sort of the sort of, I guess, philosophical motivation, I guess, for that. And then this postdoc in his lab had done this experiment that was even more like along the types of things that I was curious about. It was called the Neutral Path Project. Okay. And what he did was he took one of these naturally occurring ribozymes that cleaves itself from a virus, and of another ribozyme that had been evolved in vitro from random sequence, and that performed a ligation reaction. And what he did, like, they were totally unrelated, right? I mean, one was sort of made up, right? But what he did was he introduced mutations either one or two at a time. So just a very slow, continuous walk-through sequence. Okay. But each time it made the sequences of the two RNA enzymes more and more similar.
I see.
And eventually, they are the same sequence. And that sequence can perform both of the functions, not quite as well as either of the parents, but it's like there's only a very small, it can still do both activities, and there's like only a very small little glut in between. It's only like 4 or 5, polymorphisms that you need to do to go, whole hog on one activity to whole hog on the other activity. Wow. And the secondary structures are completely different and everything. And so that was really cool, and that really inspired me. And that really, aside from all the other sort of phenomenal aspects of MIT that made me excited about going there.
Yeah.
And that made me excited about going there.
That's really beautiful. It's directed evolution, right?
Yeah.
And it's getting at a core property of life then, right?
Yeah.
Like would that be pre-biological? What do we call biological life?
Yeah. Well, I mean, so this is something that sort of, it's useful for me to think about in sort of the aging field as well. But when you get into that field of like the RNA world and sort of origins of life, and there are a decent number of groups who study that sort of thing. That sort of idea of what is life and how can it be defined?
Yeah.
Um, and I think like in the past, sort of even in high school, you get like a definition for what is life, but it's not really a definition, right? It's like composed of cells,
Right.
can
It's a description.
self-replicate, yeah. It's like here are all the properties that most life has.
Yeah.
And that's fine, and pretty much like if you're a biologist, you're not generally that confused about whether the thing you're studying is a living thing or a non-living thing.
Right.
But but when you get into origins of life, then it's a little bit more contentious.
Yeah.
And sort of, if you have an enzyme that can replicate itself, but you have to give it all these things and sort of make conditions just right, does that really count?
Right.
So and just skipping ahead, like I think in the aging field, there's a similar issue of like when you talk like what is aging, what are the problems of aging? You can list examples of it, and but it's a little bit harder to define. And sort of deeper questions like, is anything that changes as a function of time, is that really what we're talking about as aging or not? It's sort of, it's not really, for most people, it's like working in most fields, you don't need to have that sort of nuance about whether it is aging or isn't aging, but you start to work in that field, and the sort of, well, I know it when I see it kind of explanation that works 99% of the time is no longer can be a guide all of the time.
Yeah.
So anyway, origins of life though. So that's what my I guess this was all being motivated by this question about sort of changing fields and sort of what drew me into grad school. But that was one of the things that I saw with my Ph.D. advisor, because he's an extremely successful scientist and he's very well known. I think at this point though, most people would who have heard of him sort of casually wouldn't even think of him as studying RNA enzymes. They'd think of him as studying microRNAs.
Okay.
And so I joined his lab pretty shortly after the lab had moved into studying microRNAs. And he sort of, from the beginning of that field, he was one of the really prominent people in the field. But
yeah, because that's pretty new, right?
Yeah. So this was the first discoveries were in the 90s, but at that time it was really just an example from C. elegans. And then there was sort of later in the 90s, there was like a second example also in C. elegans of a gene that worked that way. And then it was 2001 when a series of papers came out sort of saying this is not just like two genes in C. elegans. This is a whole set of genes that operate this way and function this way. Right. And so then that sort of exploded as a field, and it's connected with the phenomenon of RNAi, and that was also sort of being discovered and appreciated right around then. So so he became a really prominent person in that field. But even for him, the sort of the fact that it was a change in fields was I could see as being a difficulty in the lab and in the short term in terms of funding, because all the grants were to work on RNA enzymes. Yeah. And now it's like, okay, you have to go through another round of writing grants. And, ultimately he was successful with that, but there was a lot of uncertainty because it's like, well, and I think it helped that the field was a relatively new field where there weren't other competing experts, but it was even more stressful when it started to be like, okay, what are these genes doing? We're going to study their function. And a lot of that has to do with like developmental biology. And there really are established people there. And so, to sort of pivot from an RNA biochemistry lab to a sort of, developmental biology lab, at least in part, is a big shift. And, I think, he's a sort of prominent enough scientist where he was able to navigate that.
Yeah.
But for sort of your average everyday scientists, you can sort of appreciate like, yeah, if you're not already at that sort of level of prominence, there's a lot less freedom to do that. And if you're not, if if you're more anonymous to the sort of committee reviewing your grant,
Yeah.
they might be a little bit less trusting of funding you going into sort of a new area. And
Mhm.
so, that's sort of, what I, what I think about, or one of the things that I, sort of got this impression that the funding really does create this culture of people sort of studying the same thing
Yeah.
for long amounts of time. And there's some great counterexamples, but I think that's still like the sort of
The
the
norm.
overall theme. Yeah.
Yeah. Do you
And
think that's problematic?
I do. I think it's, there, I mean, there are advantages to it, and certainly
Mhm.
some people should do that, but I think it makes it harder to follow the science.
Yeah.
So, if you're studying, and a lot of people, if they're thinking of themselves as like a cancer biologist, right? And there's some gene, some protein that they think it might be really important to cancer biology,
Mhm.
right? Then they can go study that and they dig in.
Yeah.
If the product of like, a decade's worth of work is that this thing isn't actually relevant to cancer biology,
Yeah.
then it's like, well, what is it relevant? And, I'm still going to study it because this is the sort of the pathway or the protein complex that I study where
Yeah.
I'm the expert.
Right. You
And I'm not
become an
going to,
expert.
yeah. And I'm not going to, be able to move, even though you're already thinking of yourself as a cancer biologist, you haven't been sort of studying that broad field, right? You have a more narrow set of expertise, and you sort of say, okay, well, this is, even if this turns out not to be as important for this phenomenon of cancer biology, like this is still the thing that I'm going to study. And since all of my attempts to connect that with like higher level physiology have been to cancer biology, still probably going to be studying how it relates to cancer biology, even if it's not that important. I don't know. That's just my opinion, but that sort of seems
Yeah.
like where a lot of people can get sort of stuck in ruts.
Yeah.
Um, and it's sort of a gamble, right? So if you are the expert in something that sort of at the higher level the world then becomes really interested in, then you're golden, right?
Right.
You're the expert in that, and that's really useful to have people who sort of
Mhm.
have that in-depth knowledge of something, so
Yeah.
that when it sort of becomes important to some problem, that's the person to go to. But most people, and I think that's one of the other things I realized sort of seeing um, bigger labs as I went through my career, and seeing that there's like they're writing Nature Cell Science papers all the time, on a regular basis. These labs are great, but they're big labs, and sort of you know a lot of the people in these labs are working on projects that are risky,
Mhm.
and that are just not going to pan out,
Yeah.
and they're going to have trouble in their careers
Yeah.
because of that. But the lab is going to be fine,
Mhm.
because for every 3 people whose careers go nowhere because their projects barely even produced a paper, you're going to
Mhm.
have one that was a Nature Cell Science paper, and you have a big enough lab where So that's it's an unfortunate part of the game, but
Yeah.
yeah.
It's a very uncertain career path to take. It's very trepidatious, and there's not many fields like it anymore. I think it's where um, sort of the employee has to assume so much risk,
Mhm.
and the employer is kind of insulated. Because in the industry, it tends to be like the company will take risk and will take calculated risk, and sort of the employees are along for the ride, but it isn't as much one, I think, taking advantage of the other in some respects. So I don't know how sustainable that is long term, because these are very educated, capable people that could contribute, I think, in society in many ways, but they're making this bet that is it's starting to seem less and less worth it in the end.
Yeah, well, and but I think all these people do end up making contributions in one way or another.
Mhm.
I think the thinking about then trying to like practically get a job. There is an advantage then. I think looking at industry jobs to having some diversity in terms of what you've worked on because industry I think does sort of hire according to a need in a particular area. And so if you only have one particular area, you have to like wait around for someone to see like a need in that. So the more diversified you are, the more likely you'll see an opportunity that you can match up with. The downside though is that then if you have like a bunch of sort of diversity of experience, then in that one area where you saw an ad where you sort of match up with what they want, there's going to be other people out there who don't have that same kind of diversity, but who are more of experts in that area or just published more in that area.
Yeah.
So it's I guess there's probably some game theory to that. I'm not sure which is which is better. I mean, one thing is, so I had not worked on aging before I came to Calico. So I've in grad school I sort of I went to Dave's lab interested in studying, and I studied for a while RNA enzymes. Eventually I also switched, and I don't have any papers on RNA enzymes. I have papers on microRNAs.
Okay.
But then for my postdoc, I went to UCSF and worked for Joe DeRisi, studying infectious disease and trying to use deep sequencing technologies to discover new viruses. And so that was sort of another field, right? So different from what I'd done as an undergrad or as a grad student, or as either of the things that I'd done as a grad student. So that was a little, different. So yeah, it's
How did you make that choice? Because again, there's some considerations there, right? When you finish up a PhD, you could apply to industry in some cases, right? Or you could be thinking academia. Postdoc is the typical path. So what kind of factors were you weighing there?
Yeah, so I mean, I was really thinking about postdoc, and I sort of, I have to admit, since I joined my PhD lab, sort of wanting to work in one field and ended up working in a different field. That was a little bit of, sort of opportunity and necessity, with just sort of the way projects were going and the sort of opportunities for new projects. And so, I didn't feel like that sort of established or sort of connected to that field. And so I definitely was like open to and even favoring the idea of sort of branching out into a different field. But the other thing, there was a continuity, which was that and this is one of the really lucky things that happened. I've had great luck in a few areas, but one of the really lucky things was being in Dave's lab and sort of looking for new projects and sort of having just started to learn how to do computer programming and bioinformatics at that point gave me the opportunity to work with some of the first next generation sequencing data sets that were produced. Because if you were following that technology at that time, so this is sort of mid 2000s, sort of 2005, 2006.
Yeah. This is coming off the Human Genome Project, so all these new technologies are coming together.
Right, but they weren't really coming together yet. So they were they were in pretty bad shape. And they had super high error rates, and you could barely, you could only get through a few nucleotides before they would just crap out.
Oh, geez.
And so there were people who were thinking theoretically about like, well, this is going to be the next big thing. So how can we use this, and starting to build tools for like genomics type stuff, or for like, RNA-seq. But that wasn't really a thing yet. Most of these things could only get through like 25 nucleotides before the sequencing reaction was just over. So, but that was fine because microRNAs are 22 nucleotides.
Okay.
So that was sort of, there was like this niche area where the technology was good enough to do, now it doesn't sound like a lot, but just to put in perspective, through Sanger sequencing, the lab had sequenced about, I think at the time it was like 3,000 microRNA cDNA clones over many years of work from multiple grad students all combined. That was sort of the biggest. And then like one reaction on one of the new sequencing machines would give you many hundreds of thousands to millions of sequenced cDNA clones.
Yeah, that's wild. Yeah.
And so, so getting to analyze that, and then you start to need computer programming to actually do it. Like that was a great thing. And so I was really fortunate to be in that situation. But like you were saying, like the technology, this was before the technology was great, but it was evolving quickly, and it was clear that it was going to be great. And people were starting to experiment with ChIP-seq and with RNA-seq of mRNAs, and and with trying to do whole genome sequencing with it. And so it was clearly a technology that was going to be able to do things beyond what I had been able to do with it. Yeah. And that's where that's what sort of got me attracted to Joe's lab, where they had been doing a project for a while, because Joe is probably the central person in the development of microarray technology. And what he had started to do at UCSF was try to apply that to infectious disease research. And at one level, there was this idea of like, if you had a microarray that instead of having like mRNAs from some organism as probes, where you can look at gene expression, if you just had probes on there, and you could fit it on there against like every type of virus that is known to man, then that could, on the one hand, be sort of a diagnostic tool that is going to be a lot more efficient than trying to do an RT-PCR against one thing at a time.
Right. Yeah.
But what they had also seen is that in some cases where they had samples where the virus in the sample was unknown, if it was related to any of the viruses that were on that array, then if it was close enough, it would cross-hybridize enough. And so they'd be able to pick out it's this type of virus, and then they could use that information to go in and get the genome of the virus and sort of fully characterize it. And so this was right at the stage where his group was starting to use deep sequencing instead of microarrays to discover new types of viruses. And so that's sort of, so I sort of stayed with that technology, but moved into like a new area of science.
So how conscious was that, I guess, internalization, that this was a technology that was here for the long term? Like, how many people around you agreed with that sort of perspective? How sure were you of that?
Oh, I don't think I had no special insight in thinking that. I think everybody pretty much agreed with it. If anything, the hype was starting to be sort of in the other direction. I mean, one of the things, the work with microRNAs and RNAi, I think really helped that industry and that technology, because a lot of the other applications were still a ways off and still took a lot of work to get them, even if they could be done, to get them to work effectively. What the microRNA work really did, I think, is motivated institutions to buy machines.
Yeah.
And, because like it's sort of, you don't want to buy a machine that's like, well, this is going to be, you know, this is going to be great in like 5 years, right? I mean, academic institutions, if you're going to spend, a large sum of money on a machine, you better get a Cell Science or Nature paper out of that pronto.
Yeah.
And, and this is why I was so lucky in those very early days, because the microRNA field still wasn't that old. So there was still a lot of hype about microRNAs in general. And all of a sudden it was like, almost every, I worked with, a pretty limited number of data sets. Really 4 data sets gave me 5 papers.
Oh, wow.
And like all in top journals. So it was almost literally like every time you do an experiment run on this machine, you get a high profile paper.
Yeah, it was just so rich in novel information.
Yeah. And it was a little frustrating that then people expected that. People were like, I did deep sequencing of microRNAs. Where's my Nature paper kind of thing. But, it was like, you have to you have to discover something in that set of data. But it was, that sort of, it was a lot easier to make an important discovery sort of quickly. And, it just made things that were, very difficult to detect before, all of a sudden they're just obvious, right? It didn't even take that much insight to really figure out what was going on or to see them. And that really meant that a lot of institutions started to buy these machines. And that meant that once they had them, they could use them for developing other aspects of the technology, like ChIP-seq or sort of more general case RNA-seq. Like there started to be more of that. And it, that really, having that sort of set of early publications and that demonstration that like, this is not an abstract concept of this is going to be useful. Like it's an abstract concept that you'll be able to use it for all this other stuff down the line. But right now you can get return on your investment for buying these machines. So that really sped that along. And so I was, I was very fortunate to be sort of right in that early, an early person in that gold rush. But but I was not unique in thinking that that's in appreciating the breadth of impact for that technology.
So, so how long were you at UCSF, and when did you start thinking of your next step?
Yeah, so I was there for 6 years, and I basically had 6 years worth of funding. So I finished my job at the very end of 6 years, and then had like 2 weeks break before I started at Calico.
Oh, geez, wow!
But there was, I really started thinking about industry. I'd say in the, maybe in my 4th year, and part of it was, this is sort of, I think this like sort of changing fields, and this was also sort of even within the lab, my postdoc lab. I didn't publish as quickly as one would maybe want if you're gonna be pursuing academic jobs. And I had a backlog of like really high impact papers that are definitely helpful for getting those types of jobs, but they were all in like a different field than what I was currently working on. And so, I actually didn't apply to traditional academic jobs, which had always been my long-term career plans had always been long-term academic career path. But it was pretty clear to me that that I was not going to probably have many options, because I sort of, I didn't have a thing that was my thing. And even in my, in my postdoc, I was mostly doing sort of technology development
Mm-hmm.
projects.
Mm-hmm.
Um, so, some people were really successful sort of coming out of that lab if they like identified a new type of virus, and they're like the only person in the lab who studies, or in the world who studies that type of virus, then
Mm-hmm.
that's sort of their thing.
Yep.
And there's more work to be done, and uh, people didn't appreciate, I think, the importance of studying novel viruses then as much as they do this year. But uh, but nonetheless, it's
Yeah.
like that it sort of established you as like, this is sort of my thing, and especially if you had worked on viruses before, it's like, yes, this is a clear expert in this area.
Mm-hmm. Mm-hmm.
And maybe I would have been more successful than I think, but I didn't have confidence to sort of you know set up, like, I felt like my proposal for a lab would be like a new research direction
Yeah.
that was not derivative directly on what I'd been doing before.
Yeah.
And I don't feel like that's sort of what's being looked for in academic job searches. Um,
Yeah. I think that's unfortunate, and that's not a unique story. I've like sort of like the best trainee in arthritis. I think generally she was under, like, she won all the awards, had great publications, went to all the big postdoc labs, at least in Canada. Um, and her criticism when she was applying to these faculty jobs was that she did too many different things. Like, she started in as like a basic biologist doing like histology and stuff like that. And then she decided to switch and learn more about epidemiology and sort of population level problems and more the public health perspective, because this is, it's a disease that has a big burden on the healthcare system. And then she went and did next generation sequencing,
yeah,
which was novel in the field. But you know, I think she ended up postdocing for 6 or 7 years as well. But like, she felt it was a strength for her to understand her disease of interest, which was arthritis,
hmm
to take all these different perspectives. And I think
yeah,
now that she has a job, she is set up to pull on all that diverse knowledge.
yeah,
But it was a real uphill battle in trying to convince academics that that was a valuable route to pursue.
yeah. And I mean, the other aspect of it too is, and I think this is just the nature of academic job searches. Like, even though I'd say that I was pessimistic about my chances of getting a job, I did have enough sort of high impact science that I'd published where I'm pretty sure I could have gotten a job.
Yeah.
And I think people sometimes make sort of overplay how hard it is to get an academic position. I'm not sure it's as hard as people sort of make it seem. But one of the reasons is because if you really want to have a good chance of getting an academic position, you have to be willing to go to the whatever academic position happens to be open that
Yeah.
matches up, where the department wants someone that matches up with what you can bring,
Yeah.
which is going to be in some arbitrary corner of the country or world.
Yep.
And so there's sort of, if you want to, you know, feel good about your chances of getting a job, you cannot feel good about, like, if there's anywhere in the world that I want to be, like just a place that I enjoy or a place where I have friends.
Yeah.
If you don't care where you're going to go, then you can definitely get an academic job.
Right.
If you care about being in a certain area, even a sort of broad geographic area,
Mhm.
then there aren't very many research institutions in any area. I mean,
Mhm.
even places like Boston or the Bay Area, which are
Mhm.
notable for for how many academic institutions we have, you can still count them on your fingers.
Yeah.
These are not like, so if you have like if you have a 2% chance at each place, you're not going to have a good chance of getting anything in that area. You're going to have to
Yeah.
broaden it out. And
Yeah.
I'd sort of grown up in Southern California and went to college in Northwestern in Evanston, Chicago,
Mhm.
um, MIT, so Boston, and then Bay Area. I moved around and sort of started my life over a number
Yeah.
of times.
Yeah.
And so that was also, and my family was in Southern California, so it's I didn't want to leave the coast, but even extending it across the coast, that doesn't add that many institutions.
Yeah.
And one of the awesome but also difficult things about the California is that we have a very large number of phenomenally excellent institutions. In other words, phenomenally competitive institutions,
Yeah.
right? So
They're not the easy ones to get a job at because everyone's knocking on their door.
right.
Yeah.
So that's like that was also part of the the thinking, and so I started to look at industry jobs, and that wasn't what I had ever wanted to do, but part of it was that I think I didn't appreciate until I started really looking into and applying for industry jobs how much diversity of industry jobs there is.
Mm-hmm.
And maybe that was influenced by the sort of, the academic jobs that, sort of my peers were applying to pretty much all look about the same, right?
Yeah.
It's like,
yeah.
it's like you're going to have different amounts of space allocated to you, a different size startup package.
Yeah.
Sometimes there's some early funding that goes along with it,
Mm-hmm.
but there's not like that much sort of nuance.
Mm-hmm.
Looking at sort of industry jobs, it's like the types of jobs, what they're looking for, and what the experience would be going to like a big pharma versus sort of like a small startup. It's like those are very different types of experiences.
Yeah.
And even small startups, it really depends on what they're gonna be working on.
Yeah.
And it's and I think Calico is even like another sort of orthogonal.
There's some weird
vector.
hybrid thing. Yeah.
And so it's it's great, and I think the one of the things that, when I started looking into this, Calico didn't exist yet, and so
you were one of the first employees, right?
Yeah. So I was employee number 8, and I was really the thing that really attracted me to Calico from the outside and helped me sort of get in the door, I think, is the people that I knew who were affiliated. And when I joined, I think like everybody who was there was fantastic, but there were really only 2 people that I really knew, and that was David Botstein, who's now my boss, and he had been the co-Ph.D. advisor of my postdoc advisor at Stanford. So
wow, okay.
And he had actually done a brief little sabbatical at UCSF where I'd gotten to meet him a little bit before he went to Calico and before I applied to Calico. And then being at UCSF, I also knew Cynthia Kenyon, and was sort of knew her a little bit better, and just like the 2 of them are both they're very different, but they're both inspirational figures. And one of the things that they both really have, so there's this idea that Calico was going to do a lot of basic research,
right?
And that's something that you hear a lot, I think, in industry pitches.
Yeah.
But it's it's not always easy to take seriously. And I think of examples where it's sort of that was the pitch, and then like you see what happened to the company afterwards that like did not go at all
right.
in that direction.
Yeah.
But Cynthia and David are both sort of such strong supporters and have a deep appreciation for the practical value of basic scientific research. And what really is, when I first heard about Calico, I heard that Cynthia was going to be a consultant. And when I heard that she was leaving UCSF, where she had been for her career, and where she's a sort of a very powerful figure, that she was going to leave UCSF to go to Calico, and that David was going to be leaving Princeton to go full time. I was like, that's
Big.
not just like that's a commitment, and the company had hardly any, very few other people in it at that point. It's like that's a like it's hard to believe that this place won't actually follow through on having a commitment to basic research with those people.
And like Genentech had a pretty good notoriety for doing research, right? Where art was coming from.
Yeah. Although I have to admit, I didn't. I wasn't thinking about that that as much. It was really it was really the in retrospect, that was a good thing that I could appreciate. But but I mean, I had never. There were a number of people who had come from Genentech who were at Calico, and as I got to know them later, I could see that, but I didn't know any of them coming in the door. So it was really it was really Cynthia and David who got me excited about Calico.
Nice.
And and I think it's it wasn't so much. So I think aging is a fascinating field, and one of the things that I love about it is that there are such sort of really fundamental questions that still remain to be answered. And I've gotten to work on some of those sorts of projects. But it wasn't, I mean, one of the things that came out of this sort of moving from field to field is that the sort of, maybe this ties up the conversation nicely, but I'm not I'm not someone who's like has the one thing that I think is so important that I want to study. I really, I love the process of science and the scientific method, and using it to solve arbitrary problems. And I think the problems of aging are some of the better arbitrary problems that you could have. But it's really just the process of science that I love, and that I saw a great opportunity to do at Calico. And I guess the flip side of that is that having done so much basic research, very little applied research, the appeal, and since I hadn't worked in a particular area, I think for Calico, where they wanted to focus on basic research, and it was early days in the company, so like what aspects of the vast field of aging to focus on were still clear. I think that probably that helped make me a little bit more appealing to them as a candidate. So there was that that nice fit, I think.
Yeah. So like, how did you find out about this opportunity then? Was it just you saw a posting and you applied, or was there a little more of a inside track that you heard of?
No, I mean, I heard. So I heard from my boss, Joe, and he was the one who told me that because since he was I think he was the department chair at that point for the department that Cynthia was in. So he told me a little earlier than it was announced that she was going to be leaving UCSF. And and he told me, because he knows David so well, he told me about David, and that really, that's so he sort of brought it up with me, and that was exactly what I needed to hear to convince me that that's that's something that I should be looking into.
Yeah. And like, did you have a formal interview process and things, or did generally people understand where you were coming from and how you could contribute?
No, I did. I don't want to say too formal, because since it was so small, there was sort of an informality to everything. There weren't a lot of processes set up, so I had rounds of coming in and talking to people, but it was, it wasn't super structured. It wasn't sort of, you go through this level and then we're going to have, it was fun. It was a lot of opportunities to have some pretty interesting conversations about what could be and what would be.
Yeah. And so, it sounds like you were excited about an opportunity to do something new that was
Mhm.
sort of undefined, and but you had the space to follow what was important, or the track that came up organically, right? Um, and for me, I think um, I find that super compelling as well, and a big reason why I came here. But the main thing is that the mission or the goal of the company is so grandiose, which could seem crazy in many people's eyes, especially back when it started. But the approach is so proven in biology and in, I guess, biotech, that there's substance to it that will anchor it in the fundamentals that'll keep it going. And it allows for that exploration, that sort of wide-eyed curiosity, the redefining of things we think we know, or exploring stuff we thought we'd never be able to find out, alongside of that concrete work around the experts, some of the best scientists in the world. So it has a nice tethering, a nice grounding to it that I think is an amazing balance, and why I think it'll work in the end. Whereas I think some endeavors in industry, in longevity, seem a little, or a lot more poorly defined,
Mhm.
without a specific direction, and that can kind of spin out, I think. So I think it's a really compelling place to be. Um,
Yeah, I enjoy it, and I think I've gotten I think I've gotten an even better version of than what I hoped for.
yeah, that's
Um,
amazing.
so and I mean, I think about it in terms of uh, the things that I've published on and worked on while I've been at Calico, um, have been following the science, and
Yeah.
sort of each project being informed by the previous project and what I learned from it in terms of where to go. But it hasn't always stayed in sort of the same area.
Mhm.
And so I've published a paper on the human genetics of longevity,
Right.
with ancestry. Published a paper on naked mole rat
Yeah,
lifespan
so cool.
demographics.
Yeah.
Those that's sort of a good example of something where you know if I had been in academics, making a jump from one of those to the other, um, you know, would have been, like I said, would have been like not feasible.
Yeah.
I think, or would if I had been like a, sort of a already a famous scientist,
Right.
at like one of the top institutions, it would have been difficult, but I probably could have done it. But even if I was a lot more famous than I am, you know the the idea of making those types of big moves in terms of what I study that early in my career would not have been, as the tenure clock is counting down, like that would not have been a good idea,
Right.
right? I
Not
mean,
advisable.
like in both of those papers from Calico, had a nice impact,
Yeah.
but they're not, and to me they're sort of thematically connected,
Hmm.
but it's not in sort of an obvious practical way that I think would make it appealing to maybe a department thinking about
Yeah.
tenure, or so that's I think that's sort of that ability that I had seen, something that I value about science, of being able to sort of work on the next interesting problem. And, if you learn that this is not the thing that, that is going to give you the answer that you want, move to something that is,
Yeah.
and, learn that field and get into it. That's a rare opportunity that is hard to find both in industry and in academics. And
Right.
so I think that's something that I feel really fortunate about.
Yeah. And I would agree. I think it takes a lot of bravery on behalf of, I guess, the founders and management to allow the employees to follow the science, follow the important questions, whether they have to cross fields or not. And really, it seems that management is there to help facilitate those questions being answered. So if you need help, you need to pull in other resources and people, then that's what they'll do. And I agree. Like I haven't been here too long, but all that people have said to me has been the truth and more. So, yeah, I think it's great. And I'm really glad that we're getting to work together. And it seems more and more over the time here. It's been an absolute pleasure. And
it's a lot of fun working with you, Matt. And it's been a lot of fun chatting with you today.
Yeah. And thanks so much for doing this, for, spending your weekend time where you should be relaxing, talking to me.
This is very relaxing. I've been very relaxed. So.
Good. But this was super enlightening to me, and I've had a great time. So thank you so much.
Thank you, Matt.
All right. This has been Bridge to the Bay with Dr. Veras, with more to come.